Hey biologists, Mr. Fott here. Today we're going to be talking about tonicity and osmoregularity. First up are the must-knows. So we're going to be talking about these external environments and how they can either be hypotonic, hypertonic, or isotonic compared to our internal environments of our cells. We'll also get into water and how it moves by this process of osmosis from areas of high water potential to areas of low water potential. Additionally, we'll be referencing growth and homeostasis. Some of our characteristics of life are needed to be maintained by this constant movement of molecules across this membrane. Last but not least, this osmoregulation helps maintain water balance and allows our organisms to control their internal solute composition / water potential. So first off, we're going to refresh our memory off of this whole osmosis process. So by definition, osmosis is simply talking about the diffusion or the moving of our water particles. So at any time, if you're talking about other things besides water, it cannot be called osmosis. So osmosis is just referencing water. This, if you recall, water is one of those molecules that has the all-access pass. It can go back and forth, and nothing is going to go ahead and stop it. So this is going to be part of our passive transport. There is no energy, aka ATP here, is going to be required. There are, however, some of those proteins, those aquaporins, if you remember, which can help facilitate this transportation of these water molecules across our selectively permeable or semi-permeable membranes. So our different types of concentrations, we can use these three terms you may recall from standard bio: hypertonic, hypotonic, and isotonic. The first part of each one of these, the prefixes, is going to give you a clue as to what it's talking about in terms of concentration, solute compared to solvent. So water is known as the universal solvent. Ergo, whenever we have something else dissolved in there, it's referred to as the solute. And so we can use hyper, meaning lots of solute. So we have more solute than water. Hypo, which means less solute compared to water. And iso, which would then be referred to equal. So the diagram down below, we can see that the left-hand side happens to be hypotonic. There are less particles of solute on the left compared to hypertonic, the one on the right. There are more of those yellow spheres that we can see. So comparatively, one side is hypo, the other side is hyper. Water wants to move from an area of high concentration water, low concentration solute, to more concentrated solute, less concentration water. It wants to equilibrize, if that's a word. It wants to equalize. There we go. And so what we call this, or refer to this, is the net movement. Obviously, water particles can go back and forth, but where's the big push? Is it going to have a big push left, big push right, into or out of our cells, as we're going to reference here in the next coming examples? So in our different types of solutions, how do these play a factor into this whole homeostasis business, and how does this play out into a living organism? So for instance, salt water. If you happen to have, let's say, a shellfish, or if you have other saltwater animal species or plant species, that they are going to potentially need to do this osmoregulation. They have to balance their internal salt conditions or solute compared to their external environment, because they live in a giant solution. And so if they happen to be placed in a hypertonic solution, for instance, the outside environment gets too salty, there's too much solute, then what happens to these animal cells is that they begin to shrivel. They will wither away. And unfortunately, that organism probably will cease to exist. And so that's not a big thing that obviously we want to happen. Plant cells are a little bit more different. A great example of this plasmolysis is that if you take a look at plants and you forget to water them for a couple of days, they don't just like immediately go to dying, right? They go to wilting, this kind of in between. So when you start to, when these plants start to run out of those internal water conditions that they're normally used to, having lots of water, they start to undergo plasmolysis. And that cell, the membrane, begins to shrink. But notice how the cell wall did not change. So that's something that's different about our animal to plant cells. Sorry about the bell. That the cell wall plays a factor in this, and that the cell wall itself will not collapse. Just the membrane, which is on the inside, that part will shrivel up. Again, when these are placed in a hypertonic solution compared to their internal conditions. Animal cells don't like to be hypertonic or hypotonic. Actually, they like to be nice in between. They want to be isotonic. Plants, on the other hand, they do want to be in hypotonic solutions, which is why we need to keep watering them. So here's the flip side. If we were to place them into hypotonic solutions, for instance, freshwater organisms like this bacteria referred to as a paramecium. So this, if you happen to place it in too much water, if it were to be placed into pure distilled water, for instance, that water is going to want to rush into the cells. And if this happens, the cells will then burst or they're going to then lyse. So these cells, when placed in hypotonic solutions, if they happen to gain too much water, they will undergo this process of lysing. And if you recall, to lyse means to split apart, or in this case, they will then burst. And that obviously is not a good situation. Plants, as I've kind of already mentioned, that they want to be in this hypotonic solution, which is why we need to constantly keep watering our plants, or they need a readily available water supply. Otherwise, they will undergo plasmolysis and they'll begin to shrivel and to wilt. So plants like this, they will not lyse or explode, again, in part because of the cell wall. It's going to keep it intact. So this is called turgid, is what plants actually want to be in. Other organisms, such as the paramecium, they can actually help regulate this by using what's called a contractile vacuole. If you recall, a vacuole is kind of like a storage container, and one of the things it can store is water. And so these contractile vacuoles, they will then pump, meaning they need to use energy, the ATP thing, to then move the water out. They can actively spit out water, if you will. But again, they do require that ATP to then to try and move that water from inside to outside. They're going against the concentration gradient. Normally, as I've already mentioned, that animal cells, they want to be isotonic, in which there is no net movement. So again, water can move in and out. It's going to kind of flow freely. But where's the big push? Is it going to have a big push in, a big push out, right? Animal cells, this is where we want to be at. Plant cells, on the other hand, they do still want to be at that hypotonic solution, although I suppose they could survive if they're isotonic. But for the most part, they do like to have those hypotonic solutions. Now, if we're talking about which direction is the water going to want to move into, let's take an example right here. So if we have what's called a dialysis tube, or dialysis bag sometimes they're referred to as, and this is going to model our cell. And so it is a semi-permeable membrane. Things can move across the membrane, but specifically only water, because again, semi-permeable, it's going to be selective about which things are able to cross. Water is on the all-access pass list. So if we look and see the inside contents of the cell, we see 0.03 molar sucrose and 0.02 molar glucose. So we have two things that are dissolved within the cell. If we're to total those up, we actually get to a total value of 0.05 molar. Now, in this case, we're talking about solutes in the previous slides. It doesn't matter what it is. It could literally be anything. So all we're going to do is combine the two different concentrations, 0.03 and 0.02, to come up with our 0.05. Additionally, we compare the external environment of the cell. So we have 0.01 molar of each of the sucrose, glucose, and fructose. Again, add them all up, and we can see this is a 0.03 molar, because again, that solute concentration, it doesn't matter what it is. We're just talking about things that happen to be there. And each one of these do not have the ability to cross the membrane. Only water does. So if we do a comparison between these two, which one has a higher concentration of the solute versus a lower concentration of the solvent here. So we can see that the cell is a higher solute. So this is actually hypertonic is going to be the cell in this case, compared to the environment that it has 0.03 concentration. So this is going to be hypo, and things want to work with more water to less water, from high concentration to low concentration. Again, this diffusion process going with the concentration gradient. So if we identify it in this fashion, it's going to move from hypo to hyper, which is beaker, or the environment, into the cell. So this would cause water. Sorry again for the bell. This would cause the water to then rush into the cell. So the cell would begin to get larger. It would gain water. Potentially, if it's too much, it could possibly burst and lyse and go and undergo that process. So the next thing we want to talk about is osmoregulation, into how we're able to then balance this, right? So undergoing this process of using either contractile vacuoles, things can move in and out, maintaining different environments, hyper to hypo to iso, so that external environment can then play a factor into how our cells are going to regulate those water concentrations. And so if we need to actively pump using our active transport, for instance, to push solutes into certain areas of our body so that we can then create different environmental conditions, that is a type of thing in which AP would potentially ask questions. So how could we then change certain solute concentrations going against concentration gradients, again, active transport, to cause osmoregulation, the movement of water, to then balance out so that we have more isotonic conditions. All right, so we could go one additional thing. Instead of just talking about concentrations of things, we can actually calculate what's called the water potential. Now, there's not very many calculations in AP Bio. There are a handful of them. This happens to be one of them. They will give you the equations. The test itself is not very calculation heavy, so if you're worried about calculation portions, we will practice them and talk about them, but just know that they're not super calculation heavy, although they do have a few. More so what they want you to do is to be able to interpret the results. So if you get the final answer to the equation, that's more important that you talk about like what that is and what that means to the living organism or the question that they happen to be talking about. So this water potential, which is another one of our Greek symbols called psi, Psi is what it is. This, it kind of looks like a pitchfork almost. This is about the potential for water to move across the membrane. And we can again use internal versus external conditions to talk about different water potentials. And as mentioned in the must-knows, and just like diffusion, we want to move from areas of high water potential to areas of low water potential. A lot of these values that we're going to be seeing, they do work into the negatives. So you have to be careful. So if we have a negative value, again, we want the higher concentration, so closer to 0, versus lower concentrations, which would be more negative. So be very careful about the sign, because sometimes that will play a factor. So we have a few symbols that we can break down. Each one of those are psi, or different water potentials. And specifically, there is not just the water potential, which is the calculated value. We can use what's called the solute potential as well. That's psi sub s. So this we can use as osmotic potential, sometimes referred to as, but how much, whatever it is that's dissolved in there, how does that play a factor into whether water is going to move internal or external to a given cell? Last but not least, we have pressure potential, or the physicality of the pushing of those molecules that are then dissolved within the solution. There are two values that they will ask you to memorize, and they will expect that you have these. They will not give them to you. So these two, I would star, highlight, circle, underline, whatever you got to do. I don't ask you to memorize much, but these are one of those things. When talking about water potential, pure water, when in an open container, has a pressure potential of 0. And the MPa stands for megapascals. That's going to be our unit here. Next is going to be for plant cells. A typical plant cell is going to have, again, a pressure potential of 1 MPa, or megapascals. So those are two constants that whenever asked in any of those questions, those are the values they expect that you already know them. So once again, star, circle, highlight, underline, whatever you got to do. Pure water in an open container has 0 pressure potential. Plant cells will have a pressure potential of 1. And basically, that's what the whole water potential system is actually based off of, is either pure water, meaning our 0 scale, and plants, which is typically what we're talking about when asked questions about water potential. And they're going to be a total value of 1, so kind of 0 to 100 if you think of it like that. So what we can do to calculate this, and this is the equation they're gonna secondarily give you. They'll give you both of these. Is the solute potential. We can use what's called the negative I crit. That's sometimes the little acronym here. So it is a negative value. Again, as I mentioned, sign. You have to be very careful about whether a number is positive or negative, and it will change your answer. So this I is what's called the ionization constant. So when you dissolve things into water, it will then split apart into its ions. So how many of those ions? Is it going to be 1? Is it going to be 2? How many different charged particles will it split apart into? C is going to be our molar concentration, and that's a capital C, mind you, not a lowercase c, which would be something else. So the molar concentration, so molarity, if you recall from standard bio. R is going to be a constant value. They're going to give you this value, but I would jot it down in your notes, and again, one of those memorized values, but odds are they're going to give you that value. 0.0831. Whenever you see R, this is the number we're going to use. T is going to be the temperature, and we must be very specific about this. It has to be in Kelvin. So if you recall, again, from chemistry, how to do that conversion, take your degree Celsius, add 273. So they're going to give you questions in which they're going to give you the temperature in units of Celsius, and they're going to expect you know how to convert that to Kelvin, so you can go ahead and use this negative I crit to calculate the solute potential. And basically what this means is that if we add solute, we add more particles being dissolved. This is going to lower the solute potential, which means it's going to get more and more negative. Therefore, we're going to decrease the water potential. And it's going to kind of have an opposite effect in that we think, well, if it's decreasing water potential, then wouldn't it lose water? Actually, it's going to do the opposite. It wants to gain water. Because if you recall from the last slide, we want to move from higher water potential to lower water potential, which kind of plays a factor and makes sense into our example when talking about molarity concentrations, internal versus external conditions, hyper, hypo, iso. That the more we dissolve, the lower or more negative this water potential becomes, which means water wants to rush in there to, again, balance it out. We want to try and get this equilibrium between our internal and external conditions. So here's a few examples we'll reference here. As we've kind of mentioned a few times, first, that this psi value or water potential wants to move from the higher value to the lower value, the more negative side here. So be careful about your signs. Oftentimes you'll have two negative values for these different conditions. We want to move from the higher to the lower. A low solute concentration to the higher solute concentration. Again, the more you dissolve, the higher the solute concentration, the more water wants to move in there. From hypo, low concentration, to hyper, high concentration. Additionally, pressure could play a factor into this, and they want to physically push those particles. So if you have a high pressure system, it's going to move towards a low pressure system. These images down below kind of help demonstrate that each one of those little spheres representing some particle that's dissolved. It doesn't matter, once again, it doesn't matter what the particle is, it just matters that something's there, which is why they're not really identifying them. They're kind of generic usually, or they'll just say sugar molecules. So it could be glucose, galactose, maltose, lactose, pick your favorite sugars, it doesn't matter. They're all going to contribute the same. If you have more of them, it's going to have a negative water potential, which means water wants to rush in that direction. So we can see in this other example on the right, we have salt water versus fresh water, that the water of the movement here could use pressure to force it across that membrane additionally, from the higher pressure to the lower pressure additionally. Here's kind of another example. If you ever wondered how plants are able to then transport this water through their xylem and their phloem so that they can then get it all the way from the roots all the way to the tippy top, which would be the top of the shoots there, that the top here, the atmospheric pressure actually comes in at about a negative 95 value of our megapascals. That's hugely negative, right? Which means water wants to move in that direction. Again, from high pressure potential to low pressure potential. Then we can see kind of in between over at the leaves, they have a -8 down in the root systems. Now we're getting higher and higher. Be careful, these are negative values. I cannot stress that enough. So we have a -6 in the soil as a -3. So we have a higher water potential down at the base, which is in the soil and the roots, and a lower pressure potential once we get to the top of the plant here, which drives the water additionally with our cohesion and adhesion properties, that it can then move through those xylem and phloem so they can transport from the base of the plant all the way to the top of the plant. This last slide here is kind of summarizing a lot of those different things. So you can pause, take a look at this, kind of refresh your memory over all the different things. Please let me know if you have any additional questions. We will for sure get some practice with this. I want to spend a moment with this just to kind of talk about these different things and the equations thereof. Thank you all for joining us. Sorry this is a little bit longer, but I hope this helped to try and break things down. Have a fantastic day. Please let me know if you have any questions.